Hello, this presentation is a review on a research study of the effects of maternal immune activation on gene expression patterns in the fetal brain.
Viruses, bacteria and parasites are all pathogens which can trigger an immune response. When this immune response is triggered during pregnancy, it's referred to as a maternal immune activation, or MIA for short. From past experiments, scientists have hypothesized that MIA could be linked to autism and schizophrenia due to the increased transcription of genes which produce cytokines. The elevated levels of cytokines could then have an effect on areas of the developing fetal brain, which may cause the fetus to develop illnesses which can affect their cognitive functioning.
This experiment was conducted using mice. Use of mice proved to be essential in maintaining consistent results. Baby mice in these tests have shown to display similar symptoms of autism and schizophrenia. Different numbers of impregnated mice were randomly assigned to one of the four groups as illustrated by the diagrams. A control experiment involved injecting mice with saline alone to compare its effects with the different treatments. An important thing to note is that all mice were killed on day 12.5, although not all were injected on the same day. The flu virus treatment group were injected 3 days before the other groups, due to allowing time for the development of a flu infection. All embryonic brains were then removed and quickly frozen in tubes. In the transcriptome analysis part of the method, RNA was isolated from fetal brains and the quality was assessed using a bioanalyzer. RNA was pulled into a single sample to reduce gene expression variability not related to MIA exposure. PCR method ensured 2 samples to be obtained from each pregnant mice. RNA was then used to generate cDNA. Quantitative PCR system was used with various crystalline primers. All primers reported amplification efficiency over 90%. The last part of the method involved weighing individual placentas from each embryo. Placental weight was assessed across the treatments with a group-wise two-tailed t-test.
The microarray analysis resulted in 256 genes differentially expressed in the flu module, 294 in the poly-IC module, and 195 in the IL-6 module. Majority of the genes altered in poly-IC and IL-6 treatments were upregulated. Of the transcripts with the most changed expression in all 3 treatments, 41 genes were altered in both IL-6 and flu treatment, 31 in poly-IC and flu, 17 in poly-IC and IL-6. However, 12 genes were similarly changed in all 3 MIA modules.
As you can see from the previous slide, 5 of the 12 genes that are upregulated in all 3 of the MIA treatments belong to the crystalline gene family CRYAA, CRYBB3, CRYBB1, CRYBA1, and also CRYGB/CRYGC. Now, in order to validate this finding with an independent method, what the researchers did was that they produced a cDNA from pooled RNA from the embryos in each dam. To inquire about a possible effect of embryonic location in the uterus, RNA was separately pulled from embryos residing in the left and the right uterine horns, so there were 2 samples for each dam. For a more detailed view of the crystalline family of genes, we also tested the expression of CRYAB, CRYBA2, CRYBA4, and also CRYGC/CRYGA. What they found was that the qPCR data is highly correlated with the microarray data, insofar that the flu had an R value of 0.82, the poly-IC had an R value of 0.84, the RIL6 had a R value of 0.89, meaning that there was no obvious effect of the position in relation to this. Further qPCR analysis using the RNA from the individual flu-exposed embryonic brains indicated that the flu exposure does not equally affect all exposed embryonic brains within each dam, which could potentially contribute to eventual behavioral stability.
All 3 MIA treatments evoked strong gene expression changes. A decrease in placenta weight also had a strong correlation with these changes. All MIA treatments strongly upregulate the crystalline gene family.
Due to the activation of the crystalline gene family in response to the MIA, the crystalline gene's upregulation is an attempt to protect the fetal brain, trying to stop the damaging effects of external environmental source. However, due to the overexpression of these genes, the brain's balance of new cell growth, or neurogenesis, and differentiation is stunted. This means that as the crystalline genes down-regulate to normal levels and the development program of the brain resumes, the formation of the cortex is altered irreparably, leading to permanent changes in neural chemistry and neural interconnectivity. This causes the permanent changes in behavior in the offspring as observed in this experiment. It can also be argued, however, that this cascade of events is due to an environmental source which contributes to the risk of neurodevelopmental disorders which include schizophrenia and autism.